Welcome to this lecture series on eukaryotic gene expression, basics and benefits. Today we are going to discuss about eukaryotic protein expression systems, part 1. This is the 30th lecture in this series. In the previous lecture we had actually discussed about prokaryotic expression systems. Although this lecture series is about eukaryotic gene expression, basics and benefits, I discussed about prokaryotic expression systems at length in the last class because a number of eukaryotic proteins have been expressed in prokaryotic expression vectors and they are available as drugs and biotech drugs in the market. And as you will see in this class, the first choice of recombinant protein production is always to use a prokaryotic expression system. Because of all the expression systems that are currently available today, it is the prokaryotic expression systems which are least expensive and most economical way of producing recombinant proteins. So, it is only after you ascertain that the prokaryotic expression system is not the right choice for you that you go and choose a eukaryotic expression system for making your recombinant protein. So, it is very important to understand the prokaryotic expression systems very well. So, that is why in the last class we had discussed at length the various plasmid vectors, phage vectors, cosmate vectors that are used for not only for basic research to understand gene expression and gene regulation, but also for making a variety of recombinant proteins of interest to human and animal health. So, today let us now start looking at eukaryotic expression systems and this is, as I said, this is the number first in this series of lectures. So, I have divided this entire talk on eukaryotic protein expression systems into at least 3 or 4 different parts. Today we are going to talk about protein expression in yeast and insect cells, and in the next class we are going to talk about protein expression in mammalian cells. Following that we are going to talk how expression vectors have been designed to make recombinant proteins in plants and farm animals. And then ultimately in all these lectures we are going to discuss about cloning a gene of your interest into either a E. coli vector or a yeast vector, insect vector or a mammalian expression vector, make the protein in large amounts, purify the protein and then use it as a therapeutic. But in the final lecture in this series we are going to talk about what is called as a human gene therapy, wherein instead of making your therapeutic protein of interest in another organism or in cell lines, purifying it and giving it as a therapeutic drug, why can't we express the gene of your interest directly in the human body, so that the protein of our interest is made right inside our body. So, there is no need for you to make a recombinant protein elsewhere and then administer it as a therapeutic into the humans, but let us introduce the gene right away, express the gene right away in the human body and see whether you can get the therapeutic effect or therapeutic benefit. This has tremendous benefit in the areas of cancer therapy, in the area of treatment of genetic disorders and so on and so forth. So, after we understand how protein expression is made in various eukaryotic systems, we will now trying to understand how genes are directly being introduced into humans, various tissues of humans using viral as well as non-viral vectors, and what are the implications of this new area of research called human gene therapy for the benefit of mankind. Now, as I told in the number of classes in this lecture series, that one of the very clear benefits that came out of understanding eukaryotic gene expression is our ability to introduce genes into any cells of our choice, any organism of our choice, and then express the corresponding protein in large amounts, purify it and use it as a therapeutic. This is one of the major benefits that came out of recombinant DNA technology as well as our understanding of gene expression both in prokaryotes as well as eukaryotes. As I have listed here, for example, a number of protein or biotech drugs or biopharmaceuticals which have been produced using this kind of a recombinant DNA technology. A number of blood factors like clotting factors like factor 8, factor 9, which has tremendous implications for treatment of patients suffering from hemophilia, have been produced using this recombinant DNA methods. Thrombolytic agents like streptokinase, tissue plasminogen activator have been produced using this kind of a protein expression systems. A number of hormones like insulin, glucagon, growth hormone, various gonadotrophins have been produced. Several hematopoietic growth factors, erythropoietin, variety of Golgi stimulating factors like GCSF, GMCSF, CSF and so on and so forth have been expressed, purified and being used as therapeutic agents. A number of interferons again have tremendous applications in antiviral therapy. Interleukin based products such as interleukin 2, a number of vaccine proteins, the surface proteins of a number of viruses have been expressed in E. coli as well as yeast systems and is being used as vaccines Examples are hepatitis B vaccine and a vaccine for cervical cancer, namely the human papillomavirus antigens have been expressed in yeast systems. A number of monoclonal antibodies have been expressed and many of them are actively being used for treatment in various cancers. And in addition, there are a number of miscellaneous products which have been expressed, and basically the strategy has been to take the gene of your interest, put it in an expression vector, introduce this expression vector into the organism of your choice, make the protein in large amounts, purify, and then use it as a therapeutic product. So this, our ability to engineer genes and then engineer their expression into organism of our choice has led to a huge biotechnology industry for making biotherapeutic drugs. So the recombinant DNA therapeutics or biotherapeutics as they are called today is a huge industry by itself worth over 32 billion in the year 2003, and it is now worth much more in the year 2010. It is estimated that there are more than about 110 companies which are specializing in making this kind of recombinant DNA therapeutic, protein therapeutics for human as well as animal health. More than 80 therapeutics are in various stages of clinical development, and at least 73 biotech drugs have been approved by various regulatory agencies and are being marketed for human use. So this has tremendous implications. That's why we need to understand how today, for example, you want to clone a gene and express a gene of your interest. How will you go about? Will you choose a prokaryotic system? Will you choose a eukaryotic system? And if you choose a eukaryotic system, what kind of eukaryotic host you will choose for your purpose? I just listed some of the top 10 therapeutic proteins that have been sold in the market using this kind of recombinant DNA technology and recombinant protein expression systems. I will not go through one by one, but a number of very useful proteins have been expressed by cloning these genes into appropriate expression vectors and making these recombinant proteins in large amounts. And in fact, the recombinant protein therapeutic markets is estimated to be about $52,000 million in the year 2010. So it's a huge industry, has grown thanks to the efforts of people like Paul Berg, Herbert Boyer, and Stanley Cohen. It is these people who initiated this or developed this technology for cloning genes into expression vectors and then expressing these genes in large amounts. I have again listed some of the commercial recombinant biologicals which are available today in the market. A number of proteins have been expressed in E. coli expression systems. A number of proteins have also been expressed in yeast, either Saccharomyces cerevisiae, Pica pastores. We will discuss more about this today. They have also been expressed in a number of mammalian cell lines. So you need to know, for example, if you want to make a protein in large amounts, which of these expression systems you would choose. So we will discuss some of these things in this class. Once you decide to make a recombinant protein of your choice, will you choose a prokaryotic system, will you choose a yeast system, will you choose an insect expression system, or will you choose a mammalian expression system? How do you go about? What are the advantages and disadvantages of each one of these expression systems? So bacterial expression systems, which we have discussed very well in the last class, has a number of advantages. One of them is that these bacterial cells grow very quickly. Their doubling time is less than 20 minutes or even less than that. So you can multiply the cells in large amounts, so you can make your recombinant protein in large amounts in huge bacterial fermenters. Their yields are very high. You have very powerful bacterial promoters, so you can make your protein in large amounts. Usually something like 50 to 500 milligrams of your protein can be expressed per liter of the culture. And the media that are used for growing these bacterial cells are very, very inexpensive. So the cost of production becomes very, very low, and as well as the fermenter costs. So there are a number of advantages in producing a recombinant protein in bacterial expression systems. But there are also certain disadvantages. For example, very rarely you can express your protein in large amounts if your protein is huge. Usually if your protein size is more than 50 kilodalton, it is very, very difficult to express this protein in large amounts using a bacterial expression system. And many a times when you express certain eukaryotic proteins, they are toxic to the E. coli cells or the bacterial cells, and therefore they die. So certain eukaryotic proteins cannot be expressed in prokaryotic expression systems, and the prokaryotic machinery cannot produce disulfide bonded proteins. So if your eukaryotic protein is a highly disulfide bonded protein, so it has number of SS bonds, then it is very, very difficult to fold these proteins appropriately in a prokaryotic host and make a biologically active molecule which is properly folded and properly disulfide bonded proteins. So in such cases you have no other option but to go for a eukaryotic expression system. And the other very important drawback of a prokaryotic expression system is that the prokaryotic expression systems cannot glycosylate proteins, and they also cannot do what is called as a post translation processing of proteins, like they cannot cleave a signal peptide which is required for secretion of the proteins. Like insulin, for example, insulin is actually made as pre-pro insulin in our body, then the pre-pro part is cleaved and then you have the insulin, right. These kinds of a proteolytic processing cannot be done in a prokaryotic host. I will give you one example. For example, if you for example have a protein which is supposed to be glycosylated, and when you take this gene and put it either in a prokaryotic expression vector and a eukaryotic expression vector, and in a prokaryotic expression vector you will only see a non-glycosylated protein, and if glycosylation is essential for the biological activity of the protein, then this protein produced in a prokaryotic host is of no use for you, because it will not have the therapeutic effect. On the other hand, when you expose the same protein in insect cells which are eukaryotic cells, you can actually see a glycosylated form of the protein is now present and which can be biologically active, and therefore you have achieved your objective. So depending upon what is the end product that you are looking for, whether it has a highly disulfide bonded protein, whether proper disulfide bond has to be formed for the protein to be biologically active, or does it have to be proteolytically processed properly, which does not take place well in the prokaryotic host, or if it requires specific glycosylations, and if this glycosylation is very important for the biological activity of the protein, then such proteins need to be expressed in a eukaryotic host and not in a prokaryotic host. So let us now see what kind of eukaryotic expression systems are available, and in this class let us see how you can express these kinds of proteins in yeast and insect cells. The yeast expression systems has a number of advantages, because just like bacteria they are also single celled organisms, so they grow very quickly just like bacteria. They also give very high yields. There are certain yeast species like Pichia pastoris which can grow to very high cell densities, so you can produce large amounts of protein per liter of the culture. Again, just like bacterial culture media, the fermentation as well as the cost of the media components are very inexpensive, so the cost of production becomes very low. You can express large proteins, which is a major drawback in the prokaryotic expression systems. Many of the yeast species do have machinery for glycosylation of the proteins as well as proteolytic processing. So certain proteins, if they require glycosylation, and if the glycosylation is important for the biological activity, such proteins can be expressed in yeast species like Saccharomyces cerevisiae, Pichia pastoris, and so on and so forth. They also have certain chaperonins which help the protein to properly fold, and therefore they become biologically active, and yeast systems can handle disulfide rich bonded proteins. So protein which have huge number of disulfide bonds, and therefore they have to be properly folded. So compared to the E. coli expression systems, among the eukaryotic expression systems, the yeast expression system has a number of advantages, because they are more or less similar like bacteria in terms of cost of production and in terms of doubling time and so on and so forth. Suppose you expressed your gene in a yeast expression system, but still the protein you are not satisfied for a number of reasons, or the protein is not biologically active, then you have no other choice to try other eukaryotic expression systems. Next to yeast, the most inexpensive of the eukaryotic expression system is the insect cell, expressing your protein in insect cells, for which you use what is called a Baculovirus expression system. We will discuss this in detail. The advantages of expressing your protein in insect cells are, just as yeast, they can also express large proteins, so size is not a problem. They can also do proper glycosylation, and also they can properly cleave signal peptides and so on and so forth, so that your protein is properly processed after translation. They also have chaperonins, they help the fold proteins to fold properly once they are translated, and the insect expression systems also offer very high yields, and the media components are relatively cheap compared to other mammalian expression systems. But certain disadvantages of the yeast expression systems are, they cannot grow as rapidly as the yeast cells, so the growth is much slower, so it takes about 10 to 12 days to set up the entire system, and the cell culture is only sustainable for 4 to 5 days, and the setup time is time consuming, and it is not as simple expression system as that of yeast. So there are certain disadvantages. So if you have tried expressing your protein in yeast, you have tried expressing your protein in yeast cells, in insect cells, nothing worked, then you have no other option but to express your protein in mammalian cells. Okay. So the mammalian systems also has advantages, because again, as I said, their size is not a problem, and since they are kind of a homologous systems, since many of the cell lines used for mammalian expression systems are derived from humans or mammalian species, so they will have the right kind of glycosylation pattern, they will cleave, all the proteolytic cleavage after translation will be done exactly as same as done in the mammalian cells, and they also have proper chaperonins, and they fold the proteins correctly after translation. But the disadvantage is again it is much more difficult to set up compared to yeast and insect expression systems. Takes weeks to set up. The cell culture is only sustainable for a limited period of preterm, and it is much more difficult if the mammalian cell culture is adherent type of cells. It is much more expensive, and the setup time is very consuming. The media component, the kind of fermenter that you require, everything becomes more expensive. So when it comes to the choice of eukaryotic expression systems, you have to be extremely careful, and you need to make sure that you start from a simplest eukaryotic like yeast and then slowly go up, and if nothing works in yeast, doesn't work in insect cells, then only you go for the higher eukaryotic systems or mammalian cell lines for your expression of a recombinant protein. So prokaryotic systems are generally cheaper, but eukaryotic proteins produced in bacteria may be unstable, or they may lack biological activity due to lack of certain post translation modifications, or they may not fold properly, and therefore you have no other option but to go for eukaryotic hosts. The other important problem in expressing eukaryotic proteins in prokaryotic hosts are the bacterial cells contain certain impurities like lipopolysaccharides, certain endotoxins, and which are not acceptable if you are going to use it for use in humans. So you have to have special mechanisms to remove some of these impurities when you make these recombinant proteins in bacterial cells. So having discussed what are the choice of expression systems that is available to you, so you can either start from prokaryotic host and then gradually then try it out in yeast, then try it out in insect cells, then try it out in mammalian cells, and if even that doesn't work, you can go for plant cells, or there are also what are called as cell-free systems. We will discuss some of these things in the next few classes. So let us now focus on what kind of yeast expression systems are currently being used for making your recombinant proteins. So as I told in the many classes before, that if you decide to express your protein, express your gene in a yeast expression system, the first thing you have to do is to clone your gene of interest downstream of a yeast promoter. That is the first thing that you do. So let us now see what kind of a promoters are being used for expressing your genes of interest in yeast cells, and how do you design a yeast expression vector, and how do you introduce these genes, and what kind of expression vectors are available for a recombinant protein production in yeast cells. The two most popular organisms for making recombinant proteins in yeast cells are Saccharomyces cerevisiae, which is the common baker's yeast, as well as a methylotropic yeast called Pichia pastoris. There are also other methylotropic yeasts like Hansenella polymorpha, Aerobia lipolytica, etc. But today, for simplicity's sake, we will only discuss one methylotropic yeast, Pichia pastoris, as an example for recombinant protein production in yeast cells. So let us now take a look at what kind of expression systems are available for making recombinant proteins in yeast cells, such as Saccharomyces cerevisiae and methylotropic yeast such as Pichia pastoris. As you know, Saccharomyces cerevisiae is a very simple yeast. It is a single celled organism. It is very well characterized genetically as well as physiologically. Saccharomyces cerevisiae became a wonderful organism, model organisms for a number of eukaryotic biological processes, be it cell biology, gene expression, genetics. Saccharomyces cerevisiae served as a model system for understanding many complex biological processes in eukaryotic systems. The advantage of Saccharomyces cerevisiae is they can be grown very readily in fermenter vessels and can also be scaled up very nicely in huge bioreactors. So once you standardize your recombinant protein production to about something like 1 or 5 liter cultures, you can easily scale it up to thousands of liters of biofermenters. These technologies are very well standardized. A number of strong promoters have been isolated. They are very well characterized because yeast served as a model system for understanding eukaryotic gene expression, the role of chromatin, and so on and so forth. So a number of promoters have been studied. The entire yeast genome has been sequenced. A number of powerful yeast promoters are today available. So if you want to express your protein in large amounts, you can choose from any one of these strong promoters that are very well characterized in the Saccharomyces cerevisiae. The advantage of Saccharomyces cerevisiae is that it can carry out many post-translational modifications. So many signal transduction pathways like phosphorylation, glycosylation, protein targeting, myristoylation, they are all very well conserved in the yeast as well, which happens in the higher eukaryotes, and therefore if your protein requires any of these post-translational modifications, it can be done in the Saccharomyces cerevisiae expression system. As I said, it can be readily grown both in small and large scale bioreactors or fermenters, and more importantly, today if you are making a recombinant protein in a microorganism or in a cell line of your choice, this particular microorganism or a cell line has to be certified by regulatory agencies such as the United States Food and Drug Administration as generally recognized as safe, known as GRAS. So, only those organisms which have been certified as GRAS are allowed for use for producing human therapeutics. So, if you want to produce a human protein, you need to first make sure that the cell line or the expression system that are used has been certified as GRAS. Otherwise, even if you express this protein and even if it is biologically active, you will not be given permission to bring it out as a product in the market. So, yeast such as Saccharomyces cerevisiae has already been certified by regulatory agencies like US FDA as GRAS. So, if you make a recombinant protein in Saccharomyces, you will not have any problem in getting regulatory approvals to bring it out as a product in the market. So, this is one advantage of Saccharomyces cerevisiae. Now, let us now look at how do you distinguish between once you made your expression construct and once you introduce this expression construct into yeast cells, how do you eliminate cells which are not taken up your vector? How do you select for those cells which have taken up your expression vector? And we know in the E. coli cells or in the prokaryotic expression systems, usually antibiotic resistance markers like ampicillin, tetracycline, they are used as a selection markers. But these antibiotics do not kill yeast cells, they kill only bacteria. Therefore, the antibiotic resistance genes cannot be used as selectable markers when it comes to yeast cells. Therefore, people have developed what are called as auxotrophic yeast strains. These terminologies one has to understand. Yeast strain which can make its own amino acids or which has a normal purine or pyrimidine biosynthetic pathway, metabolic pathways are known as prototrophs. So, you do not have to provide any amino acids in the medium. If you just provide common just the salts and the carbon source, it has its own amino acid biosynthetic pathways and therefore, it can synthesize all the amino acids by itself. These are called as prototrophs. But when you take such prototrophic strains and when you introduce the deletions or mutations in one of the genes involved in any particular amino acid biosynthetic pathway, then that particular strain becomes an auxotroph for the biosynthesis of that particular amino acid. For example, if I make a mutation or deletion in the ARG4 gene, then this particular yeast strain cannot synthesize arginine. Therefore, it becomes an auxotroph for arginine. Therefore, what you now do is you now provide this ARG4 gene in a plasmid vector or a vector. So, and then put your gene of interest into that particular vector. So, when you now introduce this gene into the auxotrophic strain which cannot make arginine, all those cells which are now taken up the ARG4 plasmid now becomes prototrophs. So, they can now grow in a medium in the absence of arginine, whereas those cells which are not taken up the plasmid cannot grow in the absence of arginine. Therefore, you can select for recombinants by plating them on a media which does not contain arginine. So, all those cells which are taken up the plasmid by virtue of the presence of ARG4 on the plasmid can make arginine and therefore, can survive and grow even in the absence of arginine, right. In the same way, mutations have been made in specific genes like His4 is a gene involved in histidine biosynthesis, LUT represents a gene involved in leucine biosynthesis, TRP1 is a gene involved in tryptophan biosynthesis, URA3 is a gene involved in uridine biosynthesis. So, people have made certain mutations and these kinds of auxotrophic yeast strains have been developed, and these auxotrophic strains that are actually used for making recombinant proteins. So, you have corresponding plasmids containing all these genes and therefore, you clone your gene of interest in these vectors which contain these genes, and if you now put in the appropriate auxotrophic strains, now you can select for recombinants which can now grow in that absence of that particular amino acid or that particular nucleotide. This is how you make a selection of recombinants in the case of yeast cells. The most commonly used these expression vectors are what are called as integrative vectors or YIP, autonomously replicating high copy number vectors or abbreviated as YAP, autonomously replicating low copy number vectors or YCP, and of course, yeast artificial chromosomes. We will not discuss about yeast artificial chromosomes because these are mostly used for cloning large chunks of DNA and most commonly used for making something like genomic libraries, cDNA libraries, especially genomic libraries, and usually used for many of these mega sequencing projects. If you want to sequence human genomes or other big genomes, huge chunks of DNA are cloned into this vector, and there very rarely yeast artificial chromosomes are used for making recombinant protein. So, we will not discuss this. So, let us now see what are these various vectors and what are their distinguishing features. The YIP vectors or the integrative vectors, they do not replicate autonomously. Remember, plasmid vectors which we discussed in the prokaryotes, they contain their own origin of replication. So, they remain as extra chromosomal elements and they can multiply autonomously independent of the chromosome. Whereas, in the case of yeast, the integrative vectors, they do not have such replication origins, therefore, they cannot replicate autonomously. So, once you clone your gene into a YAP vector, you have to introduce into the chromosomal, they go and integrate with the chromosome, therefore, they replicate along with the chromosome. So, the gene of your interest is introduced to the chromosome by homologous recombination, and therefore, your gene of interest becomes part of the chromosome, yeast chromosome, and along with the chromosome, every time the cell divides, your gene also divides. So, as a result, when you clone your gene of interest to an integrative vector, your gene will be present only in a single copy. Okay? This is very important. The advantage of a YAP vectors is that the strains transformed to the YAP plasmid is extremely stable even in the absence of selective pressure, because your gene has gone and integrated into the chromosome, it is very stable. Every time the cell divides, your gene also will divide along with the rest of the genes on the chromosome, therefore, they are very stable. The disadvantage is that your gene is present only in a single copy, and therefore, the expression levels are not going to be very high. Whereas, if you are present in a multiple copy and then in extra chromosomal, since each cell contains large number of these plasmid copies or extra chromosomal copies, the protein will be produced in large amounts in that particular yeast cell. The YEP vectors, on the other hand, are episomal plasmid vectors. They can replicate autonomously because of the presence of a small 2 micron plasmid which serves as an origin of replication. So, just like the E. coli expression plasmids or cloning vectors had a E. coli origin of replication, these episomal yeast vectors contain a what is called as a 2 micron plasmid which serves as a yeast origin of replication. Therefore, they can replicate independently of the yeast chromosome. And because of the presence of an autonomous replicating unit, these plasmids can be present in very high copy numbers per cell. And most however, the disadvantage is that many of these plasmids are relatively unstable, and you have to maintain the auxotrophic marker pressure. Suppose if your 2 micron plasmid contains YEP vector contains a Lu2 marker, you have to always maintain the strain in the absence of leucine, so that the plasmid is not lost, and even under such selection pressures sometimes you lose these plasmids. Therefore, only 60 to 90% of the cells often retain the plasmids even under selective pressures. So, you cannot maintain this plasmid for too many generations. After about 5 or 10 generations, you have to go back to your parental master cell bank or the original stock and then revive and then start again. So, this is one disadvantage of the YEP vectors. The YCP vectors are what are called as yeast centromere plasmid vectors. They contain autonomously replicating sequences called ARS as well as centromere sequences, and therefore, they can replicate just like chromosomes. And the YCP vectors are typically present in very low copy numbers, about 1 to 3 per cell, and they are also not as stable as the integrative plasmids. The other very rare kind of a yeast plasmids are used called as YRP vectors. They contain autonomous replication sequences, but they do not contain the centromere sequences, and they also transform yeast at high frequencies, but they are also lost at high frequencies, and usually they are highly unstable, and very rarely YRP vectors are used. So, normally people either use the YCP vectors or the episomal vectors or the integrative vectors for most of the gene expression studies, as well as for making recombinant proteins, and among these the episomal vectors and integrative vectors are the vectors of choice for making recombinant proteins. A number of yeast vectors have been developed by various researchers across the world, either for understanding basic aspects of gene regulation, protein trafficking, cell biology, for a number of reasons, and also for making recombinant proteins in large amounts. Now, what I will do is not to discuss all of them, but just to give you a flavor, I will just discuss 1 or 2 examples of commercial yeast vectors which are currently available with certain companies, and you can just go and buy some of these vectors, clone your gene of your interest into these vectors and put them into yeast cells and make your recombinant protein in large amounts. So, what kind of commercial vectors are currently available for expression in Saccharomyces cerevisiae? For example, one of the popular vectors which is supplied by a company called Invitrogen is a vector called PYES2, which actually contains a promoter involved in galactose metabolism, namely the PGAL1 promoter, and as a result you can induce the expression of your gene by growing this by adding galactose to the medium. So, as long as you grow the yeast cells containing this particular plasmid in glucose, your gene of interest will not be expressed. The moment you change the carbon force from glucose to galactose, the promoter will be turned on and your gene of interest will be expressed. So, it is a highly regulated inducible vector system that has been developed. So, the expression of your gene can be induced by adding galactose to the medium, and as long as the medium contains glucose, your gene will not be expressed. So, basically it has a multiple cloning site. So, containing a number of restriction sites, you can clone your gene into any one of these restriction sites. Downstream of the Gal1 promoter, it also has the sequences for the proper 3' processing of the RNA such as polyrylation, so on and so forth. It also has an ampicillin resistant gene and F1R gene for growing this plasmid first in E. coli and making large amounts in E. coli, and it has a 2 micron origin for replication in the yeast cell. So, it is an episomal vector, and it has a URA3 as a selections. So, you can select the cells by plating the cells in URA minus medium. If there is no URA cell in the medium, only those cells which are taken to the plasmids will survive, whereas those cells which are not or the non-recombinants will die. So, this galactose inducible Saccharomyces expression vector is a very popular expression vector for inducing. It is a very popular inducible expression vector both for basic research as well as for producing number of proteins, recombinant proteins. Number of other such vectors are also available. For example, adjuvant cells, what are called as P escape vectors. They are a series of epitope type vectors designed for expression and functional analysis of eukaryotic genes in Saccharomyces cerevisiae. They contain 2 promoters, Gal1 as well as Gal10. So, you can clone 2 genes simultaneously, one downstream of Gal1 promoter, another downstream of Gal10 promoter, and both can be simultaneously expressed. So, by introducing one vector, you can express 2 genes. That is the advantage of the P escape vectors. The other important advantage of the P escape vectors is that they are available under a variety of oxotropic markers. You have P escape His, Lu, Trip or Ura. So, you can clone genes under any of these vector, and you can put these plasmids in all in one cell, and you can select the cells. For example, if you put all these 4 plasmids in an oxotrophic strain which is His minus, Lu minus, Trip minus and Ura minus, you can select for a cell which contains all the 4 plasmids. That means 4 into 2, 8 genes can be expressed simultaneously in a single yeast cell by using these kinds of plasmid. So, it is a tremendous advantage. So, each of these P escape vectors contains one of the 4 different yeast selectable markers, as I said, histidine, tryptophan, Lu or URA3, the same vector backbone which allows expression and epitope tag analysis of 2 different genes in a single cell. So, if you now clone 8 genes to the 4 vectors, you can put all the 4 vectors in a single cell and you can express 8 genes in a single cell at a time. This is just the map of the vector, again taken from the company website, which actually tells you here is the P Gal10 promoter, contains a multi-cloning site, and you have the 3' processing signal. So, you can clone one of your genes downstream of the P Gal10 promoter, and you can also clone your gene under the P Gal1 promoter here in the opposite direction. So, this gene will be transcribed this direction, this gene will be transcribed in the other direction, therefore, 2 proteins can be made simultaneously. Number of other features including the multiple cloning sites are given here. One can always go to the company website and get more information about these things. A number of, as I said, E. coli is one of the, Saccharomyces cerevisiae is one of the most well worked out eukaryotic hosts for gene expression as well as cell biology aspects. So, a number of E. coli Saccharomyces cerevisiae promoters have been very well characterized. I have given some of the very popular promoters which have been used. Expression vectors have been constructed using many of these promoters, such as the acid phosphatase gene promoter, alcohol dehydrogenase 1 or alcohol dehydrogenase 2 promoter, cytochrome C1 promoter, the Gal1 or the Gal1 pyridyl transferase promoter, the galactokinase promoter, the glyceraldehyde 3-phosphate dehydrogenase promoter. These are all constitutively expressed promoters. Some of them are inducible, like all the galactose metabolism genes are induced with galactose, and I also shown here some of them are inducible. This is, for example, the expression can be induced by removing phosphate in the medium. So, under phosphate deficient conditions, this promoter is turned on. So, as long as phosphate in the medium, your gene will not be expressed. Similarly, you have galactose inducible and so on and so forth. You have metallothionein promoter or the CUP1 promoter. You can induce the transgene expression by adding metals in the medium with a very small amounts of copper. And again, you have phosphoglycerate kinase, triose phosphate isomerase, UDP, galactose, epimerase or the Gal10 gene. So, you have both constitutive as well as inducible promoters for expressing your genes in Saccharomyces cerevisiae. number of recombinant proteins have been expressed using any one of these Saccharomyces cerevisiae expression vectors. I just listed some of the proteins which have been very successfully expressed, and some of them actually are available, have been licensed for use for humans and is available as products in the market. Some of the vaccines which have been very successfully expressed in the Saccharomyces cerevisiae are the hepatitis B surface antigen, the malaria circumsporozoid protein, the HIV and envelope protein. They have all been very successfully expressed, and either they have been approved for human use or are in various stages of clinical trials. Diagnostics, the hepatitis C virus protein, many HIV antigens have been expressed, and these recombinant proteins can be used for things like ELISA, Western blot and so on for diagnostic purposes. Many human proteins which have very important therapeutic benefits such as epidermal growth factor, insulin, PDGF, proinsulin, GM-CSF, antitrypsin, many blood coagulation factors like factor VIII have been very successfully expressed in Saccharomyces cerevisiae using the various expression vectors I have listed here. Now, let us spend some time having looked at the Saccharomyces cerevisiae. What kind of expression vectors are available if you want to now choose methylotropic yeast as your choice of yeast cells. Now, what are methylotropic yeasts? There are at least 4 well characterized genera called Pichia, Hansenella, Candida and Torulopsis. These are called as methylotropic yeast because they can use methanol as a sole source of carbon, whereas Saccharomyces cerevisiae cannot use methanol as a carbon source. It can only use either glucose or glycerol or fatty acids as a carbon source. It cannot metabolize methanol because the proteins or the enzymes required for methanol metabolism are not present in Saccharomyces cerevisiae. So, they harbor a very efficient and regulated metabolic pathway for methanol, otherwise toxic compound. If you add methanol, Saccharomyces cerevisiae will die, whereas these organisms can tolerate up to 1 to 2% of methanol concentrations, and they can actually metabolize methanol and use it as a carbon source. The genes coding for methanol utilization pathway are under glucose repression. So, all the genes involved in methanol metabolism are not expressed as long as you grow these methylotropic yeast in glucose as a carbon source. The moment you change the carbon source from glucose to methanol, all these genes involved in methanol metabolism are turned on. So, they offer an excellent inducible expression system. So, your transgene, if you clone your gene in the promoter of genes coding for methanol utilization pathway, as long as you grow these cells in glucose, your transgene will not be turned on. The moment you change the carbon source from glucose to methanol, your gene of interest will not be expressed. So, you can develop a very nice inducible vectors using these genes or promoters of genes involving methanol metabolism. This is briefly the methanol metabolism. The methanol is first metabolized to formaldehyde by an enzyme called alcohol oxidase, and then the formaldehyde is converted into formic acid by formaldehyde dehydrogenase, which is then converted into carbon dioxide by formate dehydrogenase, and there are many other enzymes which are not going to. These are the key enzymes, 3 key enzymes involved in methanol metabolism. And the first reaction, conversion of methanol to formaldehyde, actually takes place in an organelle called peroxisomes, and therefore, when you change the carbon source from glucose to methanol, the peroxisomes also proliferate in very high amounts in these methylotropic yeasts, and therefore, these yeasts also become very good model systems for understanding peroxisome biogenesis. So, what do you do? What are the key features of some of these methylotropic yeasts? If we take Pichia pastoris as a model system, it is a very well used or very widely used methylotropic yeast for recombinant protein production. It is a methylotropic budding yeast with a very proven record of recombinant proteins. A number of proteins have been successfully expressed in this particular organism. In the absence of glucose or glycerol, it can utilize methanol as a sole carbon source. I have already told you this. The alcohol oxidase promoter, which is the first enzyme involved in methanol metabolism, which converts methanol to formaldehyde, which is a very powerful promoter. The promoter that controls the expression of alcohol is very powerful. For example, when you grow these cells in methanol, almost 30% of the total soluble protein is accounted for by alcohol oxidase. So, the moment you change the carbon source from glucose to methanol, 30% of the total protein in the methanol grown cells is accounted for by one protein, which is alcohol oxidase. So, it has a very powerful promoter. And the other important advantage of Pichia pastoris is that the cell density of Pichia pastoris can be 10 times more than that of Saccharomyces cerevisiae. So, if you put your gene of interest in a Saccharomyces promoter and a Pichia promoter, introduce them into Saccharomyces and Pichia and grow them in identical conditions, the amount of cell density you will get in Pichia is 10 times more compared to Saccharomyces. So, per liter of the culture, you can make lot more recombinant protein if you express the gene in Pichia pastoris compared to expressing the same gene in Saccharomyces cerevisiae. So, Pichia pastoris has 2 important advantages which is listed here. One is it has a very powerful promoter called the alcohol oxidase promoter. So, your protein transgene can be expressed very high levels. Here I have just shown here. If you grow the Pichia pastoris under different carbon sources like sorbitol, glycerol, glucose or methanol, and you can see this particular band which is actually the alcohol oxidase, you do not see this band when you grow this yeast in other carbon sources. Only when growing methanol this protein is induced in very high levels, and you can see it is one of the major protein that is induced, and almost 30% of the total cellular protein is accounted for by one particular protein. So, it is a very powerful promoter and is a very abundant protein in methanol grown cells. So, this is one advantage. So, it has high level expression of alcohol oxidase enzyme methanol grown cells. The other advantage is you can see under identical conditions the amount of cells that you can obtain using a Pichia pastoris is 10 times more than Saccharomyces cerevisiae. Here are the two flasks that actually contain that shows this is the biomass generated from Saccharomyces cerevisiae, this is the biomass generated from Pichia pastoris. You can see there is a huge difference. So, Pichia pastoris can grow to very high cell densities compared to Saccharomyces cerevisiae. So, the amount of recombinant protein that you can produce per liter of culture is much higher when you use Pichia pastoris rather than Saccharomyces cerevisiae. So, Pichia pastoris has many advantages compared to Saccharomyces cerevisiae. The kind of vector that people normally use for expressing recombinant proteins in Pichia pastoris are of integrative vector type, that is the gene goes and integrates into the main chromosome, and what you normally do is that you have about 1000 base pairs of the alcohol oxidase promoter and you clone your gene downstream of this alcohol oxidase promoter. You have the multiple cloning site here, and you also have the transcription termination sequences for the proper processing of the messenger RNA. You have a oxytrophic marker, in this case the His4 marker, and you have the 3' flanking regions of AOIF. This is for recombination. I will come to it later. It also has an ampicillin resistant gene and an E. coli origin of replication. So, first you clone your gene into this vector, make this vector in large amounts by transforming to E. coli, and then make this plasmid, linearize this plasmid, and then transform into Pichia pastoris. This is the general method of expressing recombinant proteins in Pichia pastoris. So, this is what you normally do. The gene of interest is introduced downstream of the alcohol oxidase promoter, and then you linearize the vector, or and then by recombination it goes and inserts into the alcohol oxidase locus of the chromosome of the Pichia pastoris, and therefore you get a integrative type of an expression. The gene can also get into the alcohol oxidase by reciprocal recombination involving a double crossover event where the endogenous alcohol oxidase gene can be replaced by your gene of interest, and such genes are actually called as MUT-. That means these cells can no longer utilize methanol because the endogenous alcohol oxidase gene has been lost, whereas in the previous case the endogenous alcohol oxidase gene is still intact, therefore they are called as MUT-. They can still utilize methanol, whereas if your transgene has gone and integrated by replace the endogenous alcohol oxidase 1 gene, then they are called the MUT-S type, that is they are methanol sensitive, therefore they can no longer utilize methanol very efficiently, and your transgene has actually replaced the endogenous alcohol oxidase gene. In addition to the powerful alcohol oxidase promoter, there are also constitutively expressed promoters available for protein expression in Pichia pastoris. For example, the P-gap vectors contain the glyceraldehyde 3-phosphate dehydrogenase promoter, which is a constitutive promoter. It is more or less opposite to that of the alcohol oxidase promoter. It is a strong constitutive promoter. It has high expression in glucose, moderate transcription in glycerol, low transcription in methanol, which is more or less opposite to what we have seen in the case of alcohol oxidase. Alcohol oxidase is expressed in high levels in methanol, not expressed in glucose. Here it is highly expressed in glucose, but not expressed in methanol. So, again the same strategy, you can now clone your transgene of your interest downstream of the P-gap promoter, again linearize, and you can introduce it go and integrate it to the gap glycerol 3-phosphate dehydrogenase promoter site. In either of these cases, and also people have also now come up with novel markers such as zeosins. You do not have to use oxotropic marker anymore. There are now zeosin can actually kill yeast cells which have not taken up the plasmid, and you have the zeosin resistant gene. Those cells which have taken up the plasmid are now zeosin resistant, therefore you can easily select the colonies for zeosin resistance. You no longer require the oxotropic marker strain. The advantages of things like the His4 gene, which is normally used in the previous vector, and the zeosin is that zeosin is a much smaller gene, whereas His4 is a much bigger gene, and therefore the size becomes a problem when you use the old vectors can oxotropic markers, but here you get much smaller size of the plasmid inside. It's only 3.1 kb, whereas the original alcohol oxidase promoter based vector like the P-PIC vectors are much bigger. So, a number of host strains have been developed. They are either auxotrophic markers, some of them are also deficient in specific proteases, so that your recombinant protein can be successfully produced and is not protein is cleaved. I will not go into the details of it. So, a number of Pichia pastoris host strains are available for successful expression of various recombinant proteins. A number of recombinant proteins have been expressed at very high levels using the Pichia pastoris as an expression system. Bacterial proteins have been expressed, yeast proteins have been expressed, plant proteins have been expressed, many invertebrate proteins like hiruridin, which is a blood clotting protein, and many mammalian proteins have also been successfully expressed using Pichia pastoris as an expression system. Our own lab has actually used this Pichia pastoris expression system to make a recombinant hepatitis B vaccine. We have very successfully expressed the hepatitis B surface antigen in large amounts in these Pichia pastoris cells using the alcohol oxidase promoter, purified the antigen to homogeneity, and this has been given to 2 companies in India. Both of them have brought out the vaccine to the market, one as BEVAC, another as Silovac B. The other important development that took place in the case of Pichia pastoris is that, as I said, the yeast cells can also glycosylate, but many a times the glycosylation is not exactly the same as that happens in the human cells. So, when even these fine differences are very important, that means if you want to express a recombinant protein which has exactly the same kind of glycosylation pattern as seen in the mammalian cells, then people have now generated yeast strains which has the same kind of glycosylation pattern as that in the humans. These are called as glyco-engineered yeast strains. These strains have been developed by Gerngross and there is a company called Glycophi which specializes in these kinds of strains. So, this company has actually generated a library of Pichia pastoris yeast strains that have been engineered to perform specific human glycosylation at high fidelity. That means, if you now express the protein in these Pichia pastoris cells, the glycosylation pattern of your protein will be exactly the same as that happens in the humans. So, these are called as glyco-engineered yeast strains, and this company actually controls and owns over 60 patents on this glycosylation engineering the glycosylation pathway in this yeast, and therefore, you can actually produce human proteins with exactly more or less same kind of glycosylation in yeast and fungi. So, this is one major advancement that has took place in the case of recombinant protein expression in yeast cells. So, having discussed at length how you can express proteins in the yeast cells, let us now spend some time to see how you can make recombinant proteins in insect cells. Again, the principle is the same. You have to clone your gene of your interest downstream of an insect promoter. The most commonly used insect promoter for making recombinant proteins in yeast called as a polyhedrin promoter from a virus called as Baculovirus. This virus infects only the insect cells. So, let us now try to understand what is this Baculovirus and what is this polyhedrin promoter. Now, what is a Baculovirus? Baculoviruses are enveloped double-stranded DNA viruses with rod-shaped nucleocapsids. The life cycle involves 2 distinct forms of viruses, budded virus as well as occluded virus. The budded virus consists of a single nucleocapsid involved by a protein called as GP64, a virus-derived glycoprotein, and host membrane proteins, whereas the occluded virus consists of multiple nucleocapsid embedded in a protein matrix called as a polyhedrin matrix. The most extensively studied Baculovirus strain is called as the Autographa california multiple nuclear polyhedrosis virus, or abbreviated as ACMNPV, and this ACMNPV infects larval lepidopterans. So, this is the basic features of the Baculovirus, which is based on which an expression system has been developed for making recombinant proteins in insect cells. Now, what is the life cycle of this Baculovirus? It has 3 phases life cycle: an early phase, late phase, and a very late phase. In the early phase, the virus enters the cells by endocytosis, the nucleocapsids migrate to the nucleus, the viral DNA is released, and early gene expression starts. The late phase, that is about 6 to 24 hours after infection, the viral DNA replicates, the progeny nucleocapsids leave the nucleus and acquire envelope as they leave the cytoplasm, and they produce the budded virus. The very late phase, the virus formation decreases, the nucleocapsids acquire envelopes inside the nucleus to form what is called as the MNNPVs, or nuclear polyhedrosis virus, and these are embedded in a matrix made predominantly of the polyhedrin protein and forms a conclusion body. So, you can see this polyhedrin protein is actually made only during the very late phase of the virus life cycle, which is very important for the recombinant protein expression. So, this ability to make large amounts of this polyhedrin in the late phase of this virus cycle has been now taken advantage, and this feature has been used for making recombinant proteins using the Baculovirus expression system. Now, the Baculovirus expression system, abbreviated as BVS, was pioneered by Max Summers and Gail Smith in 1982. The expression system is based on replacement of the polyhedrin gene with that of a gene of your interest. So, since the polyhedrin gene is expressed at very late phase of the virus cycle and is not essential for the virus life cycle, you basically remove the polyhedrin gene and put your gene of your interest under the polyhedrin promoter. So, instead of the virus now making polyhedrin, you will now make your protein of your interest. This is the principle of Baculovirus expression system. So, some of the key features I have described here, will not go into the details. Basically what you do in the case if you want to express your gene in insect cells using Baculovirus expression system, the gene of interest is cloned into a transfer vector based on E. coli plasmid that carries a segment of the DNA from SCA and PB. So, you have a bacterial expression plasmid which contains two regions which are homologous to the polyhedrin gene of the insect. So, first clone your gene between these homologous regions, and this is called as a transfer vector, and make in large amounts in E. coli cells, and then you co-transfect this along with Baculovirus DNA into the insect cells. So, homologous recombination takes place between these regions in your transfer vector and the polyhedrin gene, and as a result you now get a recombinant viral genome which now contains your cloned gene between the polyhedrin downstream of polyhedrin instead of the polyhedrin gene. And this DNA can now be introduced in the insect cells, and now the virus recombinant virus now generated, and then the recombinant protein can be made in large amounts. This is the basic strategy of generating a recombinant baculovirus. So, the advantage of the Baculovirus expression system is that it can the polyhedrin promoter is a very highly active promoter, highly inducible promoter. Almost 50% of the total cellular protein can be your recombinant protein. Like the yeast, it also permits many post-translational modification, proper disulfide bond can be formed, and it can also do signal peptide cleavage because in eukaryotic expression system very easy to scale up because insect cells can be grown in very large amounts in inexpensive media, and also is a very inexpensive expression system compared to mammalian expression systems. I will not go to the details of this. A number of commercial Baculovirus expression systems are now available from a number of companies, like Invitrogen sells what is called a back-to-back Baculovirus expression system. The Becton Dickinson Bioscience or the CloneTech sells what is called a Bac-Pac6 or a Baculo Gold system. Invitrogen sells again Baculo Direct, and another company sells Flashback or a BacMagic, several variants, so that basically it becomes very easy now using these commercial systems to express your gene of interest using a Baculovirus expression system. I have also given here the advantages of all these commercially available Baculovirus expression systems. Again, I will not go to the details. If you are really interested in expressing a recombinant protein Baculovirus, one can go through all these things and see which one of them is more suitable to you. Go to that particular company, buy the system, clone your gene into the system, make a recombinant Baculovirus, and make your protein of your interest. There are also many insect cell lines which have been specifically developed for recombinant protein production in insect cells. For example, you have the SF9 cells which has been produced from a insect called Spodoptera frugifera. It is basically used for recombinant Baculovirus production, the intracellular protein expression, and plaque assay. You have a cell line called SF21, again derived from Spodoptera, again used for intracellular protein production as well as secretion of recombinant proteins. You have what is called the HI5 cells produced from Trichoplusianae, primarily used for secretion of recombinant proteins, and you have what is called the Tri-X, which is actually derivative of the SF9 and primarily used for intracellular protein production. Each one of them have its own advantages and disadvantages. So, once you made your recombinant Baculovirus, you can choose any one of these insect cell lines to introduce the recombinant virus and then produce a recombinant protein in large amounts. A number of recombinant proteins again have been expressed using the Baculovirus expression systems. I have just listed some of them here. You can see huge number of proteins of either parasite proteins, viral proteins, human proteins have been made using the Baculovirus expression systems. So, what we have discussed today is two major eukaryotic expression systems, namely the insect expression systems as well as yeast expression systems. And when it comes to the choice, remember an eukaryotic expression system always costs 5 to 10 times that of E. coli, and usually whenever you want to make a recombinant protein, your first choice is a prokaryotic expression systems. Only if you are not happy with the prokaryotic expression system for any reason, then only you start trying eukaryotic expression system. Even among the eukaryotic expression system, it is the yeast and insect expression systems are the least expensive, so you try them first, and if it still does not work, then only you go for the mammalian expression systems, which we will discuss in the next class. So, how do you decide on what expression system to choose? The choice depends on the size and character of your protein. If you have to express a very large protein, you have no choice but to use only a eukaryotic expression system. If you are expressing a small protein, go for a prokaryotic expression system, but if your protein glycosylation is very important for the protein, then you cannot use a prokaryotic host. So, you have to either choose a Baculovirus or a yeast or a mammalian expression system. Again, some specific post-translational modifications are very important for your protein. Again, you cannot go for a prokaryotic expression system, and you have to choose one of the eukaryotic expression system. So, the choice of expression system depends on the kind of protein that I am trying to express and the kind of modifications that the protein required for its biological activity. So, I just listed some of the important proteins which has a kind of a commercial interest. I also strongly suggest you to go and visit the websites of some of these very important companies like Invitrogen, Clonetech, Novagen, Strategene. They have put in a lot of effort to develop a number of expression vectors for both prokaryotic as well as eukaryotic expression, and if you go and there are very beautiful presentations of how these vectors work, and one can go and then educate yourself in these company websites and then decide what kind of expression vector will be suitable for your work and then use it for your recombinant protein production. I think I will stop here.